This video is going to focus on the chemical digestion and absorption of each of our 4 macromolecules, and most of this is going to happen in the small intestines, but we are going to look at some of these are going to actually start up in the mouth. And for all of these, this is going to get pretty technical. So for what I want you to kind of memorize and be ready to answer questions on for the test is you need to know the 4 macromolecules, you need to know how they break apart, and where that happens. So there's going to be lots of enzyme names and lots of smaller molecule names, and I want you to kind of weed through that. And there's a great picture on page 983 of your book, and it's kind of a chart that gives the lowdown for carbohydrates, proteins, and fats in your book. So I would absolutely listen here and go through the book and read, but that picture is a really good summary of everything that's happening. So we're going to start with carbohydrates. So carbohydrates we know is a macromolecule, and then they are made up of sugars. Now here you're going to see lots of sugar names, but realize anything that ends in "ose" is just a sugar, so don't let that freak you out. And then also the word saccharide means a sugar. So monosaccharide means 1 sugar, di means 2, and then oligo means a series of a few, not a long chain. That's called a carbohydrate, but a smaller, short chain is called oligosaccharide. So again, don't get freaked out with all of these enzyme names or saccharide names. Just kind of focus on each macromolecule and how they break down and where that happens. So for carbohydrates, that process happens in the mouth. It starts in the mouth. And the first enzyme to start breaking it down is amylase, and that's in your saliva. And so it breaks down the carbohydrate, which just means a long chain. Sometimes it's called a carbohydrate, sometimes it's called a starch. It breaks it down into shorter sections called oligosaccharides. So in the oral cavity, that amylase likes a pH of 6.8 to 7. And then the amylase breaks apart or denatures when it hits the stomach acid. And then the amylase is digested by a different enzyme called pepsin. So keep in mind as we go through this, all of the enzymes are proteins. And so that's sometimes hard to remember too, but we're going to get to that here in a little bit. So half of our starch is digested before it reaches the small intestine. And this is one of the one of the macromolecules that starts digestion in the mouth. Not only with chewing, the mechanical digestion, but the chemical digestion as well. Okay, so then as we move through, the amylase coming from the pancreas also breaks down the starches into those little sections of oligosaccharides, and that happens in the small intestines. Really not much is happening in the stomach for carbohydrates. So part of it happens in the mouth, and then the rest happens in the small intestine. So once that is broken apart into those oligosaccharides or into maltose, which is just a sugar, it also can be broken up into lactose, fructose, glucose. Those are all just individual sugar names. And so those sections of carbohydrates called oligosaccharides, they contact the brush border enzymes, which we talked about in the small intestine video, and those break apart the oligosaccharides even further. Okay, so it says converting. It's not really, weird. That's a weird word to use right there. It's just breaking apart. I always think about beads in a strand, and they're in a strand, and then they break apart into individual beads. I don't think that's converting. It's just breaking them apart. So I don't think that we need to jump in lactose intolerance here. I think that this section is difficult enough. So of that brush border, is using the enzymes to break it apart, and so the cell membrane of those absorption cells in the small intestine has transport proteins embedded in the cell membrane that absorb those sugars as soon as the brush border enzymes release them. So you can think about the enzymes kind of on the surface breaking apart these sugars, and then right when the sugar is broken apart, it's absorbed into the cell. Your book has a separate picture of this, but this description is plenty clear. So 80% of our absorbed sugar is glucose. So normally we see glucose the most, but you know, the maltose, fructose, lactose, those are other options as well. And it is transported out of the absorptive cell into the extracellular fluid by facilitated diffusion. When the sugar is in that extracellular fluid, it increases the osmolarity or the concentration, and then it pulls water from the lumen of the intestine by the process of osmosis. So if you need to review those are back from A&P1, but I'm not so picky that you can draw me a picture or explain exactly how that happens, but basically the sugar is in the absorptive cell and then out of the absorptive cell into the extracellular fluid by facilitated diffusion, and then that change in the extracellular fluid concentration pulls water by osmosis. This just talks about other sugars. Basically, once they are out of the base of the cell by facilitated diffusion, then they are absorbed into the blood capillaries. And then going through the system of blood capillaries and arteries and veins, then they are delivered them to the liver. So this is where we talk about blood sugar level. This is exactly what we're talking about. And this is how the sugar gets in the blood, and then this is how the liver is able to regulate that level. Okay, so that's all for carbohydrates. Our next macromolecule that we're going to talk about as far as absorption and digestion is proteins. So proteins is the macromolecule, and they are made up of amino acids. So once a protein is broken down, it's just amino acids, and they are absorbed in the small intestine. So nothing happens before the small intestine, and the small intestine absorbs amino acids from 3 sources. So they can come from our food. They can also come from digestive enzymes digested by each other. Okay, that sounds like a lot going on. So let's go back to amylase and pepsin from the carbohydrates. Well, we just said that amylase, once it gets to the stomach, is digested by another enzyme, which is pepsin. Well, enzymes are just proteins. So this is basically talking about that some of the enzymes that are used earlier in the digestive process are done being used. And so then the small intestine absorbs those enzymes as well. And they're proteins. And then the other source is epithelial cells that have sloughed off, and they are digested by enzymes as well. So this kind of gives you an example of about 44 to 60 grams per day is coming from our diet, and then the other 30 grams a day is coming from the other enzymes or other amino acids. Okay, you might see the word peptide, and I just want to talk about that. A peptide is technically a section of multiple amino acids that are joined together, but a little short segment. So that then when it gets longer than that, then it's when we call it a protein. And again, that picture on page 983 is a great resource of where we're going to see these words. But when you look at the word peptide and then the word peptidase, we know anything that ends in an ase is an enzyme. Well, it makes sense that a peptidase is an enzyme that breaks apart proteins. So a lot of times we can kind of clue ourselves into what's happening just by breaking apart the name of the enzyme. So they start working in the stomach. They're breaking apart those proteins in the stomach, and then it is absorbed in the small intestine. But it's also continued to be digested in the small intestine as well. Now, here's going to start listing lots of enzymes. I really just want you to focus on the main enzyme for each of these macromolecules. So this talks about breaks apart the peptide bond, and then breaks them into shorter peptides, and then moves on through to the small intestine. Okay, then we see in the small intestine, those smaller peptide chains are broken apart one amino acid at a time. And there's 3 more enzymes that do this, the pancreatic enzyme and the brush border enzymes, and where's my third one? 2 brush border enzymes, I'm sorry. So again, I don't need you to memorize these names and to know exactly which amino acid gets removed. I'm really not picky about that. Just realize that the protein digestion and absorption kind of goes in phases, and those amino acids are not absorbed until they are free amino acids or they're separated from their other amino acids that they were joined together. So this kind of shows you a picture of how the enzyme is breaking apart the peptide, and then once those amino acids are free, then they are absorbed through the cells of the small intestine and then moved into the blood capillary. So again, those free amino acids, that means they're roaming around by themselves. They are not connected to a protein anymore. So brush border enzymes finish the task. They make free amino acids and they're absorbed by the epithelial cells of the small intestine. And then the facilitated diffusion, that's the process one more time that is going to move the amino acids into the bloodstream. So again, facilitated diffusion, just to recap, is going from high to low concentration. So if we have a high concentration of amino acids in the epithelial cells, then they're going to want to move across into the bloodstream because there is a lower concentration of them in the blood, of amino acids in the bloodstream. And facilitated diffusion just means through a protein channel that's in the cell membrane. And this talks about infants absorbing proteins by pinocytosis. I'm not really picky about that. I'm going to save lipids and nucleic acids for a second video.